cholesterol. So now we have the major component, especially which you can see in the mammalian cell, which is your cholesterol. Of course, this cholesterol also is the major component of your membrane. So you should never forget, this is major component of your membrane. This controls or regulates the fluidity of your mammalian cell. So apart from this, the cholesterol will act as a precursor for the synthesis of bile acids. So you have bile acid, bile salt, for all those things, your cholesterol will act as the precursor molecule. Apart from the steroid, as we discussed in the earlier slide, the steroid hormone like aldosterone you have, and so many other steroids, sex hormones, all those hormones will be steroids only. So these steroid hormones also will be synthesized from cholesterol, and also vitamin D will be synthesized from cholesterol. Though you will be standing in the sunlight, you will be producing vitamin D, but ergocalciferol, which is present in the skin, once if UV irradiation happens to that, it will be converted to vitamin D3. Again, the cholesterol only will act as a precursor to synthesize the ergocalciferol, which is a precursor of vitamin D3. So all these molecules will be synthesized with the help of course, your cholesterol. And also already we saw that it controls or regulates the fluidity of your membrane. So apart from the regulation of fluidity of your membrane, it also favors in the insulation. So it will insulate the axon, especially all the nerve fiber in which you have the presence of myelin sheath. So the myelin sheath is just a plasma membrane which wound around your axon several times. So that wounding around of your axon several times, actually it insulates. So the presence of cholesterol in those membrane provides this insulation capacity for your myelin sheath. So whichever the axon which is having the myelin sheath, which will be insulated with the help of your cholesterol. So apart from that, you must be thinking how it gets incorporated with the biological membrane. In the biological membrane, how your cholesterol would go and get incorporated, because you would have drawn like two lines, one round shaped structure, one circle head, and you will be drawing two tails. So the tail region contributes for your non-polar structure, and the head group contributes for the polar structure. So such kind of biological structure which will be existing, how this cholesterol goes and gets incorporates itself in such biological membrane, you have to think. So now here, you just look into the cholesterol structure. You have A, B, C, D, the serine ring structure, followed by the alkyl side chain. So till carbon 27 it has, and you have this exact alkyl side chain and the A, B, C, D serine ring structure. And in 1, 2, and 3, the third position you have hydroxyl group. So this OH group, wherever you will draw O, there this O group will go. So wherever you are drawing, even in the cholesterol, you will be drawing O structure. So the O is hydrophilic structure of the amphipathic compound. So here that zero or the round structure, which is your hydroxyl group, now this hydroxyl group only will be exposed outside the membrane. The remaining, the bulky group where you have the alkyl side chain and ABCD, the serine ring, the entire structure will get buried into the lipid bilayer. So now what happens? This also will act in such a way that the head group, which is your hydroxyl group, which will act as a polar structure that can interact with the external surface or outside environment. Whereas the bulky structure, whereas this A, B, C, D and the other alkyl side chain and all, will form the hydrophobic acyl group, which can be buried inside the lipid bilayer. So this kind of arrangement, so here this region will be facing exterior, and the remaining region or the body region of your cholesterol will get buried itself into the lipid bilayer. This kind of existence will make or regulate the fluidity of your membrane. The fluidity, the movement of your acyl chains of other fatty acids, everything will be governed with the help of this arrangement where OH is exposed outside the water, so or outside or the surface of your membrane, whereas the remaining structure will be buried or incorporated into the lipid bilayer. So this is very important arrangement. With the help of this arrangement only, you can see that cholesterol will be incorporated in the biomembranes. It governs the fluidity as well as it insulates in the insulates your nerve wherever your myelin sheath is there, because of this kind of arrangement itself. So you have to draw the cholesterol structure, and you have to draw the write the numbering. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You have to close your ring structure, then start with the next ring structure, then close the ring structure, then you will be going for the other structures. Then 15, 16, 17, 18, then 19, then you will start again 20, 21, 22, 23, 24, 25, 26, and 27. The numbering will be like that. So in the third position, you will have OH group. So this is the structure of cholesterol. Sometimes in some of the books, they would have given 7-dehydrocholesterol. That is also correct. That is a little bit modified cholesterol with the help of certain enzymes, and that will also act as a precursor for the synthesis of vitamin D3. Everybody draw the structure and write only the import. Don't copy all the points. Just write down what are all the things which is synthesized, where and all it will be present, and how the arrangement will look like. So you can write this part as hydrophobic, hydrophilic or polar head group. The remaining you put an arrow mark, you put a flower bracket and write it as hydrophobic acyl